Hola, mi nombre es Alan Sánchez y voy a presentar mi situación problema de la materia de análisis y estudio de biosistemas. Dihydrofolate reductase, also known as DHFR. EC 1.5.1.3 is the nomenclature for dihydrofolate reductase. It is a monomeric enzyme responsible for the reduction of dihydrofolic acid into tetrahydrofolic acid. Each digit in the enzyme commission nomenclature has a meaning. The first one indicates the type of reaction. The second digit indicates the type of link it acts on. The third digit indicates the class of molecule it acts on, and the last digit indicates the molecule it acts on. DHFR, as said, catalyzes the double reduction of folate to turn it into tetrahydrofolate through the oxidation of two NADPH molecules, as you can see in the figure. This takes place in the metabolic route of folate biosynthesis. Because of the malfunction of DHFR causes a lack of folate, this affects the generation and growth of cells such as erythrocytes, resulting in the development of abnormalities. These abnormalities make these cells incapable of transporting oxygen and nutrients, resulting in the development of megaloblastic anemia. Gibbs free energy. This parameter indicates how feasible a reaction is and whether it happens spontaneously or not. For the formation of dihydrofolate and tetrahydrofolate, none of these reactions are spontaneous for themselves. But thanks to the cofactor NADPH double oxidation that has a Gibbs negative free energy of minus 21 kilojoules per mole, this makes the reaction spontaneous. Km corresponds to the concentration of substrate in which the speed of reaction of the enzyme is equal to the half value of the maximum speed of reaction. In humans, DHFR Km has a value of 0.006 micromoles. For purified DHFR, this has a maximum speed value of 17.5 micromoles per minute per milligram of protein produced. One of the inhibitors for DHFR is the methotrexate that inhibits this enzyme. This type of inhibition that it presents, it's a competitive inhibition, and its value of inhibition, its constant of inhibition value, it's 2.74 micromoles. A biotherapeutic consists in the application of proteins towards the treatment of some kind of illness, in this case, megaloblastic anemia, that it takes origin in an initial stage of diagnosis. For this, the synthesis of antibodies, monoclonal antibodies that take as an antigen the enzyme dihydrofolate reductase, DHFR, it makes possible the quantification of the same, of DHFR, in any given sample. The correct diagnosis of origin of the megaloblastic anemia, it allows the to give the patient an adequate treatment. To obtain this biotherapeutic product, it is important to start with the cold preparation that it takes about 10 days to produce, and its posterior clarification. From the previous sample, we know that the antibodies that are of our interest are contained in the hybridome of the sample, so we can make it go through a affinity chromatography that will separate the antibody of our interest to test later if we can measure the amount of DHFR and to determine whether it is a failure in its synthesis or in its functions to get the correct treatment. Compared to the obtainment of treatments in the chemical industry for pharmaceutics, these have a very large presence of pharmaceutical actives in the environment at alarming rates. So by using biotherapeutic obtainment or biotechnology methods, this will provide a safe for the environment and human health production reducing costs and contamination to the environment. Now that we've seen how important it is to study molecular biology and its advantages and its uses, it is important to concentrate on using that kind of methods to make the world a better place for future generations.